Okay, so welcome to this next video in which we're discussing the pathology of asthma. Okay, so we're currently on the late phase of a asthmatic attack. Okay, so we've had this secondary exposure to the allergen which causes the activation of the mast cells. The mast cells then start releasing histamine, leukotriene B4, the cysteinyl leukotrienes, and also tumor necrosis factor alpha. Histamine, leukotriene B4, and cysteinyl leukotrienes all lead to the immediate phase of the asthmatic attack, which occurs within minutes of exposure to the allergen. And now we're discussing the late phase, which occurs more within hours, okay? And this is achieved by tumor necrosis factor alpha, which goes to the endothelial cells of the blood vessels within the lamina propria, of which there will be arterioles, capillaries, and postcapillary venules, and is going to cause type 2 activation of those endothelial cells. Now this takes much longer, which is why the late phase takes time, basically. It takes hours for type 2 activation to occur, and this is because it involves changes in expression of proteins within those endothelial cells. Now, what happens is these endothelial cells change the expressions of proteins so that they can now recruit T helper 2 cells from the blood. And it's a little bit of a mystery where these T helper 2 cells actually come from, really, because we don't know which antigen they're actually targeted against. You see, you might say, oh, well, surely it'll be the original allergen that we initiated the adaptive immune response against, but that primary exposure to the allergen that caused that initial adaptive immune response, that could have happened absolutely ages ago, basically, and there might not be any of these T helper 2 cells left. Now, yes, there will be memory T cells, okay, there will be memory T cells, and those could be activated again by an antigen presenting cell taking the allergen to them and then activating them to become T helper 0 cells and then differentiate into T helper 2 cells. However, that would take longer than we have, really, so we don't know where these T helper 2 cells come from, and it does seem to be a bit of a mystery. However, what we do know is that T helper 2 cells start flooding into the lamina propria of the bronchi, and what they start doing is releasing 2 very powerful cytokines. So they start releasing interleukin-5, and they also start secreting something known as the granulocyte macrophage colony stimulating factor. So this is a fantastically long named protein. So granulocyte macrophage colony stimulating factor, and for short, the granulocyte macrophage colony stimulating factor is often abbreviated to the GM-CSF. Okay, so GM for granulocyte macrophage, and then CSF for colony stimulating factor. So this is equal to this. That's also got another name just to make things even more confusing. So it's also known as the colony stimulating factor CSF2. Okay, so you might hear it referred to as the granulocyte macrophage colony stimulating factor, or you might hear it referred to as the colony stimulating factor 2. Now, these 2 cytokines are very good at recruiting ear cinephiles to the site of where they are, basically. So interleukin-5 and CSF2/GM-CSF, they're going to go up within the lamina propria of the bronchi, and they're basically going to lead to ear cinephils, which is a type of white blood cell within the blood, being recruited from the blood into the affected area. So you're going to start getting ear cinephile recruitment. Now, this is odd, basically, because ear cinephils, these proteins are actually going to act on the eosinophils, so they're going to make a change on the level of the eosinophiles. So you don't usually recruit eosinophils, okay? So what has to happen is the interleukin-5 and this granulocyte macrophage colony stimulating factor will go into the blood, act on the eosinophils, and then the eosinophils will then be able to interact with the endothelial cells and be recruited into the site of inflammation. Okay, in addition, this interleukin-5 and the granulocyte macrophage colony stimulating factor, or the colony stimulating factor 2, they also activate the eosinophils. So let me just show you the structure of an eosinophil, okay, because it's a rarer type of leukocyte. It's rarer to actually see these involved in something. Okay, so eosinophils have this bilobed nucleus, okay, like so. And they are called eosinophils because they take up the dye eosin very, very well. So if they're stained with eosin, they will appear bright red because they take up eosin so well. Okay, and their nucleus will then appear quite dark black. So they're often compared to a sunburnt face, basically, because these look like sunglasses. The nucleus looks like sunglasses, and then the rest of the cell looks bright red, basically. Okay, so I might even colour it in bright red. So here is our eosinophil, okay, with its bilobed nucleus. And I want to emphasize it doesn't have two nuclei. It just has one nucleus, which has a very bizarre shape. It has this shape where you have these two separate lobes connected by a little tube, okay? Right, so these eosinophils are also going to be activated to produce and release certain things. So, interleukin-5 and granulocyte macrophage colony stimulating factor are going to firstly lead to the recruitment of these eosinophils, but they're also going to lead to their activation. And what do the eosinophils, once they're activated, release? Well, basically, they are full of granules. Just like mast cells, they're full of granules, which is why they're known as a granulocyte. So there are three types of granulocyte: neutrophils, eosinophils, and basophils. Okay, and all of them have their own little granules. Now these eosinophils have granules which are filled with horrendously cytotoxic proteins. Okay, so two proteins: major basic protein and eosinophil cationic peptide. Okay, and both of these proteins, both major basic protein and eosinophil cationic peptide, or eosinophil cationic protein, they are both very, very cytotoxic. So if you release major basic protein or eosinophil cationic protein onto cells, it will kill the cell. Okay, so these are horrendously cytotoxic. So these are very, very powerful leukocytes, basically. These are not to be taken lightly. They are very dangerous cells. And they're usually used if you have a multicellular pathogen to deal with. So for instance, if you have a parasitic worm or something, you can launch eosinophil attacks on them, and they just release these horrors onto the surface of the multicellular organism and try to destroy it. And they're the only cell really that has a chance of taking down this pathogen. The others haven't got hope really. Okay, so eosinophils are a cell to be respected, basically, a very powerful cell of the immune system. Okay, so this is bad news, basically. We are recruiting these horrible cells into the site of where there is no pathogen to be destroyed. They're being recruited into the lamina propria of the bronchi by these T helper 2 cells, and now they're going to start releasing their granules. They're going to start releasing major basic protein and eosinophil cationic protein. In addition, interleukin-5 and granulocyte macrophage colony stimulating factor also causes them to make more cystinoyl leukotrienes, which maintains the contraction of these smooth muscle cells, basically, at least to a certain extent, not to the same extent as you have in the immediate phase, but to a certain extent maintains the contraction of the smooth muscle cells lining the bronchi, okay, and therefore produces bronchoconstriction. So that's one of the portions of the late phase, that you continue to have bronchoconstriction. But then the other important things are that if you're releasing these horrible proteins, this major basic protein and this ear cinephile cationic protein, those are going to lead to damage to the endothelial cells. Okay, so this leads to damage of the mucosa. And remember, the mucosa was the name for the epithelial cells, the basement membrane, and the lamina propria together. So damage to the mucosa. Okay, so what ends up happening is you start getting missing epithelial cells, missing basement membrane, and the important thing that this leads to is you get exposure of neurons. So let me draw this. So before, what we had was our mucosa looked like this. We had these lovely columnar epithelial cells here, maybe with a few goblet cells interdispersed amongst them. So let's say this is a goblet cell over here in green, okay? And the columnar epithelial cells were ciliated and everything was working nicely, okay? And they were sitting on a basement membrane, which, remember, mainly consisted of collagen. And then underneath that basement membrane, you then had the lamina propria, which was this next layer of connective tissue, and this was all part of the mucosa, basically. The columnar epithelial cells, the basement membrane, and then the lamina propria. Now, after you have brought these horrible ear cinephiles in, and they've released cytotoxic proteins, then you're going to start getting the death of certain of these epithelial cells. Okay, so you'll have gaps within the epithelium now. You'll have great holes, okay? And what this can lead to is it can lead to the exposure of neurons, which are within the lamina propria. They can end up exposed, so let me put a neuron here. They can end up exposed to the airway, basically, to the lumen of the airway. So I'll put the basement membrane in here. Okay, and then we have the lamina propria underneath. So there were neurons in the lamina propria, but once you've removed the epithelium, they can now end up exposed to the airway, basically. And this is why you can now start getting non-allergic asthmatic attacks, because things like cold air, environmental pollutants, horrible chemicals in cigarette smoke, things like that can come in, stimulate this nerve, and this can now lead to the asthmatic attack. So this will lead to the activation of the endothelial cells, and therefore the beginning of the acute inflammatory response, leading to the swelling of the lamina propria, and also it can lead to the contraction of the smooth muscle cell layer. Okay, so you'll get contraction and also the swelling of the lamina propria, and this will lead to the same thing as before, but it's now triggered by neurons rather than by mast cells. Okay, so this can lead to a non-allergic or a intrinsic or a non-atopic asthmatic attack. Okay, so this is how after years and years of having allergic asthmatic attacks, you can then progress to having non-allergic asthmatic attacks, because every time you have an allergic asthmatic attack, you gradually damage the mucosa more and more. And I want to stress that you don't just go from this to this with one allergic asthmatic attack. You have them repeatedly. Every time you're exposed to the allergen, you'll have an allergic asthmatic attack, and gradually you'll damage the mucosa more and more, and this will eventually lead to the exposure of these neurons, and then stimuli such as cold air, environmental pollutants, sulfur dioxide, nitrogen dioxide, noxious chemicals in cigarette smoke. These can then stimulate these neurons, which will then trigger an asthmatic attack. Okay, so that now concludes our discussion of the pathology of asthma. In the next video in this playlist, we'll turn our attention to the drug treatment of asthma.